seventh graders. I'm here today to wish you a happy National DNA Day. And as you can see from the double helix here, right, we've got the special structure of DNA, which has become so famous. Why do we celebrate National DNA Day? Well, it marks the discovery of DNA's double helix by James Watson, Francis Crick, and Rosalind Franklin in 1953. And it commemorates their successful completion of the Human Genome Project, which was on April 25th, 2003. Something that was completed just 50 years after DNA's discovery. And why is the Human Genome Project so important? It's important because it shows us the sequencing and mapping of all of Homo sapiens' genetic blueprint, which is a lot, 30,000 genes. Okay, today to celebrate this National DNA Day, I've kind of decorated myself a little special. So in addition to my lab coat, I've got my double helix earrings on. And you can see that twisted double ladder, and I've got also my DNA bracelet on. So I don't get to wear these all the time, so a little bit of celebration here. So we're going to extract some strawberry DNA today. Yes, it's possible. Unbelievable, but it's possible. With special ingredients, we can separate the DNA out of the nucleus of the cells of strawberries using the ingredients that you will see here. We will use some salt, we will use some soap, meat tenderizer, and then finally some ethyl alcohol. Isopropyl alcohol is also okay. The ethanol seems to work a little bit better. So it's very simple, and I've got my strawberry here, and some people use a blender, but we can actually do this by hand. And we start out with 90 milliliters of water, and so it's just under the 100 mark. Okay. And I'm then going to add some soap. It's 10 milliliters of soap. Stir them together. And then what we do is we add the salt, and it calls for about 1/4 to 1/2 teaspoon of salt. And then I'll continue to stir and dissolve those three ingredients together. And normally in the classroom, I would do a cheek cell DNA extract with you guys and have you swish around some salt water in your mouth and return it to a cup and then add the detergent and actually get to harvest some of our own cheek cell DNA. So now I dissolve them a bit, and then I will add the meat tenderizer. And that's about 1/4 to 1/2 teaspoonful as well. And give them a good stirring. Now, also don't forget that in the scientific method to do an experiment properly, you want to also have a control. So I will have a control set aside in another cup that will have all the same ingredients in it except for one step that will be left out. And then we're going to compare the difference. Okay, after those are sufficiently stirred so that most of the salt and meat tenderizer are dissolved, we're going to pour the detergent and enzyme solution, that's what the meat tenderizer is, it's basically an enzyme, into this bag that contains the strawberry. And we try to remove as much air as possible. Okay. And then I'm basically just going to squish that strawberry without deflating, or rather without bursting the bag. So do this part carefully. Also, by the way, if you choose to do this at home, please be sure to ask your parents about using any ingredients that are in the fridge and that that's okay with them, so we don't run down our food supplies during this time. This can also be done with peas. It can also be done with bananas successfully, and as I said, the cheek cells. You could do it with just about any fruit or vegetable. You could do it with onions and get pretty good results. The reason why we tend to use strawberries is they just yield a lot more DNA. It has something to do with the chromosomal structure inside of the cell. Okay, so we've got this nice pinkish solution here. All right, we want to make sure we get rid of the big chunks. Okay, and let's say I'm pretty close. Okay, so we're going to do, the next part is important because you have to strain out the big chunks of strawberry. Pour this through the strainer, get most of it in the cup. And then I'm just going to use a spoon to help press out some more of the strawberry detergent and salt combination. And you get that nice kind of pink smoothie look inside the cup. I also want to mention what the salt does. The salt is important because it helps to release the DNA strands by separating the protein, that's the protein structure in there, from the nucleic acid that they are bonded to. The detergent is simply because you need a lipid to help break apart the cell membranes. So you want to get the DNA out of the cell membranes. The same for the enzyme in the meat tenderizer. It's going to help get that DNA out of the nucleus, which is surrounded by a nuclear membrane. Okay, I think we did a pretty good job there. So I'm just going to put that on the side. Right, so now you're going to see that there's a foamy layer of soap that's still on top. So what I'm going to do is just try to remove a little bit of the soap from the surface. Let's drop it in there for now. I'm going to pour this solution into the smaller cup. Try to hold back some of the bubbles. Okay, I'm just going to turn this around this way so the label doesn't obstruct your view. Okay, so if you take a look in this cup now, you'll see this really nice strawberry smoothie looking liquid. It will smell very good, but beware, it contains soap, it contains salt, it's going to be very salty, and meat tenderizer. You do not want to drink that, okay? I repeat, you do not want to drink that. All right, so now the final step is to add our cold ethanol solution. Now, normally I would use a 90% ethanol solution or isopropyl 70%, and then chill it in the freezer so it's really cold. Chill it for about an hour. But supplies being what they are, I have hand sanitizer and I don't have ethanol right now. So hand sanitizer is 70% ethyl alcohol and it works pretty well. So what we're going to do is carefully drop some of the isopropyl, or rather ethanol, into the cup that contains our strawberry juice along with the other ingredients. And we're going to squeeze it along the sides of the cup. It's probably going to be about 5 milliliters worth. And we wait a little bit. You can carefully swirl, but I do not recommend stirring for this part. You want the DNA to start appearing, and you don't want any of the remaining pulp to mix in with the rest of the solution. If you take a look in the glass now, you will see that already there are some mucus looking types of globules that have already started to form. Guess what that is? If you guessed DNA, strawberry DNA, you are correct. Now, I have also a cup that I prepared earlier. As you let it sit, the DNA strands will increase to the point where you have quite a lot of white strands. Then using a tweezers or a glass rod, okay, you actually would be able to gather some of them together once you formed enough and actually pull it right out of the solution. Is that amazing or what? We have extracted DNA from a strawberry. This is microscopic stuff, but because we were able to clump a whole bunch of it together, we are able to see large amounts of DNA clustered in one massive globule. Take a look at how this one's doing. Yeah, they're forming, and they're forming pretty fast along the sides. Here's my control that I set aside earlier. This was just some regular strawberry juice that was added to salt and to soap. However, no alcohol was added to this solution. And as you can see, very different. And this was made a few hours ago too. No changes. This one, which was just made now, already you can see the white clusters of DNA. This one was made the same time as the control, and look at the difference. So although we have a little bit of soap and a little bit of pulp in there, nowhere near the white clusters that you see in the ones where the alcohol has been added. So this is something you can do at home. In a few days, I'll follow up by getting some close-up pictures and seeing how it looks under my microscope. We are getting closer today. I will follow up with a few questions on Google Classroom to see what you gleaned from this video experience, and I will follow up with some more pictures as well. Thanks for watching, and again, happy National DNA Day. Makes you you and me me. See ya.